LSR Institute in Singapore. So I kindly request Dr. Chirag Desai to share the session of the symposium and introduce the speaker. Thank you.
Thank you very much, Senthil. And good afternoon, ladies and gentlemen. It's my privilege to be here in this symposium today. And the task given to me is on behalf of Professor Chattu, unfortunately who could not make it because of his personal reason. So I'll be opening the session, and before I do open, let me remember that when the previous millennium was about to end on 31st December 1999, we were very anxious that whether the next day the sun is rising or not, whether the flights will land on time or not, and Y2K bug had beaten very badly to all of us, and we were preparing for Y2K for end of Y2K for quite some time. But next day morning, when we got up, we saw that nothing has changed. But after almost 15 years, if I actually think that whether nothing has changed, if I think of biotechnology, I see that many things have changed. In the end of last millennium, we were talking about genomics and proteomics and metabolomics and many omics. And we had some platforms, some technology that would help us, the oncologists, in the diagnosis of the disease, in the management of the disease, in monitoring the disease and prognostication of the disease, and so on and so forth. But with unfolding of human genome, sometimes early in this millennium, I realized that so many things have changed, and the way we are looking at many things has changed. We have many more platforms like Sanger sequencing and ARM-PCR, we have NGS now, we have many sources of the tissue. We are looking at the free DNA in plasma. In plant biotechnology also we have a lot of development. So in every area, every walks of life, we are looking at biotechnology and some technology platform that is helping us in day in and day out activities. So I compliment iBioanalysis and the faculty who have come here to enlighten us in what is happening in last few years and what is going to happen over next few years. India has already established itself as a IT giant, and in the coming decade, it's our duty to ensure that we also take the similar kind of lead in biotechnology in utilizing various platforms. And I'm sure that participation in today's symposium will help all of us in ensuring that goal. So with that, let me invite Dr. William Telford. As Senthil already said, he is the associate scientist at Center for Cancer Research in National Cancer Institute, National Institute of Health, USA. His key area is flow cytometry, and he is going to tell us about flow cytometry, how it works, and what it can do. Dr. William Telford, please.
Okay, well, thank you very much for having me today, and thank you to iBioA for initiating both this workshop and the introduction this afternoon. What I'd like to talk about are really the first of several technologies that iBioA works with. I'm probably okay with this. I think I'll do okay there. Okay. A field that I'm sure many of you are familiar with, flow cytometry, how it works, and what we can do with it. Let me tell you a little bit about myself. I work at the National Cancer Institute at NIH. I'm not a professor. I'm actually a core manager there. I run a core facility, and what we try to do, in addition to helping our investigators perform their research, is also run a small internal research and development program aimed at improving both the applications of flow cytometry and also the technology. So, and this is very much guided by the scientists we serve at our institute. So what I'd like to talk about is how flow cytometers work, because I think this is something that gets lost a little bit in the application. Is there a laser pointer around, or should I just use, I can just use my pointer. Yeah, I'll just use my mouse pointer. Because unlike next-generation sequencing, where things seem to go obsolete every few years, even the older technology can still be very valuable in laboratories today. This is a FACSCalibur. It was developed back in the 1990s, and it has since given way to multi-parametric, multi-laser instruments like the Fortessa. But the principles involved are still very similar between the two, and I'd like to sort of remind us of some of those principles. So first of all, to build a flow cytometer, you need a biological sample. You need a way to get the cells in a stream passing through a laser, which will both scatter the laser light and also excite fluorochromes on its surface. You need to detect the scatter and or the fluorescence of those cells, and then you need to process that information in a computer. And I'd like to sort of remind us of some of the technology that goes into this, because we tend to often forget it as we use these instruments as black boxes. Now, the way we get the cells to the instrument is that we shoot them down a tube. But we can't just use an ordinary capillary, for example, because the flow at the edges will be slower than the flow at the center. The cells will spray out the end. So I'm sure many of you who do flow cytometry are familiar with the fact that you have to fill up a sheath tank to run your instrument. What sheath is an outer stream that pushes in on the sample stream and focuses it into the center of the laser beam. You usually do this in a conical nozzle so that the cells are aimed directly right in the path of the laser and can take advantage of that laser's excitation properties. Now, there are a number of ways to do this. The older instrument used what is called a jet and air system, where the cells were shot out into the open air. Most of the analyzers we use today are cuvette flow cytometers, where we shoot the cells through a quartz cuvette. Some instruments, like the FACSARIA, are hybrid jet and air systems, where we analyze the cells in a quartz cuvette and then send them out into the open air so that they can be sorted. But the technology is still very much the same. This sheath and core system, or hydrodynamic focusing, as it's called, is still critical in most flow cytometers. And you can actually see this on the instruments that you run yourself. This is a flow cell from a FACSCalibur, so this is an older instrument, but the same flow cell is still used in modern instruments today. What we do is we inject the sample in through a needle, we surround it with a sheath stream, and then it goes up through a cuvette, and that's where the cells are analyzed. And if you look at the instrument, you can actually see all this going on here. Your sample is down here, your sheath is entering here, your sample is being injected up through this needle, and then the cells are being analyzed up at the flow cell up here. So you can actually see this going on in the instruments that you're running in your laboratory. Now, the fluidics on these machines are relatively simple. Here is the flow cell over here, where we inject the samples into the stream. We have a sheath tank over here, a waste tank over here. We pressurize the sheath tank. The sheath flows in, it surrounds the sample stream, and then pushes in and pulls those samples into the instrument. We have 2 air pumps over here, and really this only has 4 tubes in it. The fluidics on our machines are actually pretty simple. They're not that difficult to understand, and the physical principles behind this are also not that difficult to understand. So what happens when the cells are running through the instrument is that the sheath stream is pushing in on the stream, and when you turn up the sample pressure to make the cells go faster, the cells are not actually increasing in velocity. That sample stream in the middle is actually increasing in diameter. That's why we want to run our samples at as low a sample pressure as we can to make sure that the cells are lined up nice and straight inside the flow cell. If the sample stream is too wide, we can have 2 cells arrive at the same time, and our analysis can be complicated. So this is something to think about when you're actually running samples on your own instrument. And you can actually see this. What I've done here is put a camera on a flow cell. This is a flow cell illuminated with a laser beam. I've run some green fluorescent beads through the flow cell and blocked out the laser light with the sheath stream on, and you can see how the cells are neatly focused in the center of the flow cell. If you turn off the sheath flow, the cells start to disperse throughout the entire flow cell. We're no longer focusing the cells in the middle of the laser beam, and we don't get as good an analysis. So hydrodynamic focusing and sheath is a critical element of flow cytometry. Now, the next thing we need to do is excite our cells. And we were fortunate in flow because the development of flow cytometers coincided very closely with the development of the laser. This is an ideal way to get a concentrated amount of light energy onto a very small spot for exciting both fluorescent probes that are on the surface of the cell, but also looking at the scattered light properties of the cell. Some of the earlier instruments used point sources like mercury arc lamps. These were not nearly as efficient for detection of fluorescent probes or light scatter. Virtually all cytometers today, as most of us know, rely on laser sources. And the earliest laser sources that we used were actually quite dependent on the fluorescent probes that we had available. Now, fluorescence, which is a low molecular weight fluorescent probe that almost everyone in this room, I'm sure, is very familiar with, was one of the most common fluorochromes back in the 1970s and the 1980s. It is ideally excited with a 488 blue-green laser. And fortunately, this line was available on argon ion lasers, which were a common laser available to us back in the 1980s. So because of this, almost all flow cytometers have this blue-green 488 nanometer laser on it. And if you open up the instrument and look inside, you'll see this pretty green cyan glow from a 488 nanometer laser. So what we would do is we would take our fluorescein molecule. In many cases, we would attach it to an antibody, which would be directed against a marker on the surface of the cell. There are many other physiological probes that also use fluorescein as well. And they could be readily excited on an instrument that had a 488 nanometer laser and could detect the green emission that was produced by fluorescein. And the earliest cytometers were only one or two color. We could only detect one or two fluorescent probes at the same time. Now, at the time, the argon ion lasers that we had available to us were big water-cooled monsters. They were 2 meters long. They were very powerful. They were very good lasers, but they were not very practical for flow cytometry. They required large instruments. And that's why most of the instruments that were developed back in the 1980s were large machines, like the Dako Cytomation MoFlow, the Beckman Coulter Ultra. Some of you may have experience on a Fax Vantage cell sorter. These instruments had to be big to accommodate these large laser sources. Now, lasers, fortunately, have gotten smaller. This is an air-cooled 488 nanometer laser. It's still a gas laser, but it's far smaller in size, and it could be air-cooled rather than water-cooled. So what it allowed was the development back in 1987, and I'm old enough to remember this, of the first true benchtop flow cytometer that many of you, I'm sure, are familiar with, the FACSCan. And this was a big deal back in 1987. This was a small cytometer that could be connected to a microcomputer and could analyze 3 different fluorescent probes simultaneously: fluorescein and 2 others, one phycoerythrin and one called PE-Cy5. We were also fortunate that this development of this instrument correlated with the beginning of the HIV epidemic because it allowed us to do CD4 and CD8 T-cell analysis on patient samples using a multicolor machine. So in addition to fluorescein, we took advantage of another probe, phycoerythrin. This is a not low molecular weight. It's a natural product. It's derived from cyanobacteria. And a third dye called PE-Cy5 that is actually phycoerythrin coupled to a low molecular weight dye called Cy5. Unlike FITC, which emitted in the green, these emit in the yellow and the red range. So using instruments like the FACSCan, we were able to do, for the first time, multicolor flow cytometry. We could look at 3 different markers simultaneously, and we could use filters and mirrors to separate the signals of these probes and look at 3 different markers on cells simultaneously and interrelationally. And we'll talk about a little more about that in a few minutes. Now, fortunately, laser technology has developed quite rapidly since then. Red lasers were added to flow cytometers in the early 1990s, either helium neon lasers or red diode lasers. These emit between 633 and 640 nanometer, and they can excite a variety of fluorescent probes as well. So what that allowed us to do was increase the number of simultaneous parameters we could do on a cytometer. If you had a flow cytometer equipped with a 488 blue-green and a red laser, you could not only excite FITC-PE and PE-Cy5, but you could excite another phycobiliprotein, APC, and another pair of dyes called APC-Phi5 and APC-Cy7. So the number of colors we were able to analyze simultaneously was increasing. And this was correlated, fortunately, with the fact that we were starting to identify more markers on the surface of immune cells. So we could take advantage of these additional parameters and start to look with increasing complexity at the structure of the immune system. A third laser type, violet lasers in the 405 nanometer range, were introduced in the 1990s. If you've got a cell phone, you probably have a blue LED in it. This is very much a relative of the violet laser. It was developed by Shuji Nakamura back in the 90s. And it turns out these violet lasers can excite another group of fluorescent probes, a variety of them, that can be added on to the red excited and the blue-green excited probes that we've already been using. So now, multiple lasers are normal on flow cytometers, and in fact, many instruments have multiple lasers on them. Almost all cytometers will have the blue-green 488, but many will have a red and many will have a violet as well. And what this allows us to do now is excite up to 16 different fluorescent parameters and do it simultaneously. This is very powerful because we can look at great detail at the structure, for example, of the immune system to look at B cells and T cells and myeloid cells. And there are a variety of commercial platforms out there that are equipped with these three laser sources. I call them the triad, the three major laser sources that we use. So the development of those lasers and their introduction into cytometers coincided with the development of a variety of fluorescent probes that could be excited by those lasers. The ones we've already talked about, an additional group of dyes called the brilliant violet dyes, for example, that can be excited with a violet laser. So if you have the right instrument with the right technical capabilities, you can analyze a vast variety of fluorescent probes and look at many cell characteristics simultaneously. And the reality is we now have laser technology that can excite anywhere from the UV all the way up into the infrared. This is something my laboratory works on quite a bit, and we can mount a variety of different laser sources that pretty much cover the entire visible spectrum. This allows us to excite virtually any fluorescent probe that we want, and it gives us tremendous flexibility in doing flow cytometry. What you can also see, too, is that the lasers are getting smaller. This is an air-cooled argon up here. These are two solid-state blue-green lasers and a little tiny blue-green laser down here that I can literally hold in the palm of my hand. So the lasers are getting smaller. They're still pretty powerful. We can build smaller and smaller instruments that have greater and greater capabilities. And you can go a little crazy if you want with this. Adrian Smith, who's a colleague of mine at the Centenary Institute in Sydney, Australia, has built a flow cytometer with 10 different lasers on it. Now, he's not going to use all these lasers simultaneously, but this selection allows him to excite any new fluorescent probe that comes along. If something new is discovered, a new probe, a new fluorescent protein, Adrian has the ability to test it on his cytometer. And it makes for a very powerful technology for looking at fluorescence. So we've got our nozzle, we've got our laser, we shine our laser onto the stream. We do focus the laser on the stream to get as much light energy onto the cell as possible. What you're looking at here actually is a cytometer that has 4 lasers, and the lasers you can actually see the beams crossing through here, violet, blue, green, yellow, and red. This little hole in the background is a pinhole that is coupled by a fiber optic to the detectors on the instrument. So the cells pass down past these pinholes, they're illuminated by the laser, and then any fluorescent probe on the cell, any excitation, emission from that, will pass down the pinhole and be transmitted by the fiber optic to the detectors on the machines. Now, how do we detect the fluorescence of our probes? There are a variety of ways to do it. Most cytometers use photomultiplier tubes. This is not a new technology. They've been around for decades. PMTs are very sensitive photon detectors. Big ones can detect a single photon under some circumstances. And they're extremely sensitive, and they actually make up the detection end of most commercial flow cytometers. Photodiodes are just little solar cells. They don't have to have energy or electricity inputted into them the way PMTs do. They're not quite as sensitive. They're often used to detect forward light scatter, which we'll talk about in a few minutes. But some newer instruments use avalanche photodiodes. This is a much more sensitive photodiode that behaves in many ways like a PMT. It's a relatively new piece of technology. It's much cheaper than a PMT. And at the end of my talk, I'll show you an instrument that uses APDs rather than PMTs as detectors. So this is what the detectors look like. These are two instruments from my laboratory. This is a BD influx. It's a cell sorter. I've got my LSR2 over here. And the PMTs are contained in these blocks behind the laser, behind the nozzle, and the fluorescent signals are transmitted to these PMTs where the signals are cleaned up using filters and mirrors that I'll talk about in a few minutes. There are many different ways to do this. The influx uses 45-degree angle detection geometry. The LSR2 and a lot of the BD machines that you're probably familiar with actually use these octagons and use a series of mirrors to bounce the signals to different detectors. But you can see how many detectors there are on here. We're collecting 5, 10, 15, maybe even more fluorescent signals. And we need to be able to separate them and analyze them simultaneously. Now, the optics that we use to do this, there are two main types. First of all, we use bandpass filters. These are filters that allow only certain wavelengths of light to pass through them and exclude everything else. So a fluorescein filter will have a transmission band at 530 nanometer with a 30 nanometer window. It will only let the fluorescein signal through, but will block everything else out. And all of the detectors on your flow cytometer will have a filter, a bandpass filter in front of them that corresponds to the fluorescent probe that you're interested in, be it FITC or PE or propydium iodide or whatever it is. Now that's to identify single fluorescent probes. To separate signals from each other in multicolor analyses, we use dichroic mirrors. These are mirrors that allow some wavelengths to pass and others to be reflected. So for example, a long pass dichroic mirror will allow longer wavelengths to pass through, but they will reflect shorter wavelengths. So a 530 nanometer long pass filter will let any light above 530 green through, but it will reflect everything else at a 90-degree angle. A short pass dichroic mirror will do the opposite. It will transmit the shorter wavelengths, but reflect the longer ones. So if you pull apart one of your cytometers and actually look at the filters, they'll have a little designation stamped on the side of them if they're a bandpass filter, a dichroic, or a long pass or a short pass dichroic mirror. And by arranging these things into an array, you can separate multiple fluorescent signals of different wavelengths and detect them with relatively little overlap. Now, there are two parameters, however, that are not based on fluorescence. They are based on scattered laser light from the 488 nanometer laser. And I'm sure many of you have heard of these. Forward light scatter is any light that is scattered by the cell at a 180-degree angle to the incoming laser beam. So if you put your laser over here, you shine it on a cell, you use a metal bar to block the direct laser light and look only for scattered laser light, that can be detected by a photodiode. And forward scatter correlates very roughly with cell size. The bigger the size of a cell, the higher the forward scatter signal. So granulocytes, monocytes, macrophages will have a higher forward scatter signal than lymphocytes or erythrocytes. We also look at a characteristic called side scatter. Instead of looking 180 degrees opposite of the incoming beam, we look at 90 degree off to the side, orthogonal as we call it. We use the same sort of optic. We use a blocker bar to keep the main laser light out, and any light that's scattered by the cell can be picked up by a photodiode or a PMT over here. Notice here, and for forward scatter as well, we're using a bandpass filter to make sure that only the blue-green laser light gets to this detector. Many of our instruments have multiple lasers now, so we need to keep that other light out. Side scatter correlates roughly with optical density of the cell. A granulocyte, a macrophage will have a neutrophil, will have a higher side scatter signal than a lymphocyte or erythrocyte will. So if you analyze cells based on forward and side scatter, you get the very familiar differential pattern where we can identify the red cells, the lymphocytes, the monocytes, macrophages, and often granulocytes as well, by using just forward and side scatter alone. We're not even using fluorescence yet. We're just using the scattered laser light from the 488 nanometer laser. And for those of you who do flow cytometry, you know how important this is. This is usually our first step when we analyze cells, is to look at the scattered laser light properties. Now, we often also want to detect fluorescent probes that we have attached to the surface of the cell or inserted inside the cell. So this is not scattering now. We simply want to excite the fluorochrome that we've put on our cell and then detect that fluorescence using a photomultiplier tube. So what we do is we excite our cells. We again look at the 90-degree angle, and we use a bandpass filter to filter out the light that we want to detect. So in this case, we would use a 530-30 nanometer, a green fluorescein filter, if we wanted to detect FITC on the surface of the cell. Now, if we want to look at more than 2 fluorescent parameters, we need to use a dichroic mirror to separate the signals. In this case, we've labeled our cells with green fluorescein and orange phycoerythrin. We use a 560 short pass dichroic to separate the FITC and the PE signal. This mirror allows the FITC signal to pass through. It reflects the PE signal. And we can look at those 2 parameters simultaneously labeled on the surface of the cell. And this can start to get complicated. You can use a whole series of dichroic mirrors and bandpass filters to detect many fluorescent probes simultaneously. Here we're looking at FITC, PE, and PE Psi5. We're also looking at side scatter. So we use a series of ascending long-pass dichroic mirrors to gradually pick off the 488, the 530, the 585 orange, and then the red signal, and do multi-parametric, multi-label flow cytometry. Now, how does this actually look in real machines? I'm sure many of you are familiar with the FACSCalibur. This is an instrument. I still have this instrument in my lab. It was built back in the 1990s. It is a 4-color flow cytometer. And if you open up the lid on the thing, you can actually see where the lasers are. The 488 laser, it's an argon laser. It's sitting right here. This is a little air duct that takes all the hot air from the laser and heats up your room. There's a little red laser over here. And the caliper has 3 detectors off the 488 laser and 1 detector off the red. So it can detect 4 fluorescent probes simultaneously, plus forward scatter and side scatter. We would call that a 6-parameter machine. 4 fluorescent parameters and 2 scatter parameters. And this is what the optical bench looks like on the inside. It's not oriented in 45-degree angles like I showed you before, but we use 3 dichroic mirrors, first to separate the FITC and side scatter signal, and then one down here to reflect the PE and red probe signal, and then another to split those up. So we use dichroic mirrors to split up the signals, and then we clean up the signals using bandpass filters. Now, this is a 20-year-old machine. New flow cytometers that can now analyze 10, 12, 15, 18 colors simultaneously are really not all that different. They just use more mirrors and more bandpass filters. But the underlying technology remains very much the same. And this is what one of them looks like. This is our Fortessa. This is an instrument we just had installed last fall, the BD LSR Fortessa. This instrument has 6 lasers on it. So it's got the traditional 488, it's got the red and the violet, so it has the 3 lasers that are really important. We have a green, a blue, and a UV laser on it to excite certain fluorescent proteins and other specialized fluorescent probes that we need. So it has 6 lasers and 6 intercepts. 1, 2, 3, 4, 5, 6. This is the flow cell here. These are all the laser beams, and the little holes behind the laser beams are where a fiber optic is sending the fluorescent signals to the detectors on the instrument. So you can see the red lasers here, the violet lasers here. This is the ultraviolet laser over here. It's more complicated, but really there is just more material inside than there is in a simpler machine. Here are the detectors. They're hidden, but under each of these little black caps is a photomultiplier tube, a PMT detector, and the bandpass filters and dichroics are contained in this little circle inside the sets of detectors. So what actually happens is the signals are reflected off these mirrors and transmitted at lower and lower wavelengths to these detectors. Each laser has a set of PMTs. And it's a little complicated, but this is the optical layout of the instrument. But it's really not that bad. We have a detector assigned to each fluorescent probe that we want to analyze. We have bandpass filters, and we have dichroic mirrors. The same principles on a simple 2-laser instrument are being applied here. And this instrument can do a great deal. It can detect 18 parameters, fluorescent parameters simultaneously, and it can detect a wide variety of exotic fluorescent proteins and other fluorescent molecules. Now, I won't talk about it today, but some of you, I'm sure, have done fluorescence-activated cell sorting on many cytometers. After you analyze the cells, you can then physically separate them into tubes. These instruments are equipped with a nozzle that can vibrate. When the cell is interrogated and analyzed, the stream is then broken into droplets. The droplets are then assigned an electrical charge, and the droplets divert into tubes. We call this fluorescence-activated cell sorting. So remember that all sorters are flow cytometers, but not all flow cytometers are sorters. Most of the cytometers that we have out there do not sort. The more advanced ones do. But not everybody needs to sort, and analysis is often good enough for many of the things that we do. I won't talk about this much today, but do keep this technology in mind. It's really critical, a critical aspect of flow cytometry. Now, what are some of the things that we analyze by flow? Some of the applications, now that we've talked about some of the physical technology that goes on behind the instrument. And I'm sure many of you have done at least some of these. Some of you have probably done all of them. Cell surface labeling is probably the most common application for flow cytometry. There are dozens, if not hundreds, of different fluorescent probes out there that with different excitation and emission properties that can be used for flow cytometry. These can be attached to antibodies or fusion proteins or other reagents that can home to extracellular receptors on cells, intracellular targets, other things as well. And remember, one of the great things about flow cytometry is that we're not limited to looking at only one aspect of the cell. This is a simple analysis where I've labeled cells with a fluorescein anti-CD8 antibody and a PE, which is our other fluorochrome, anti-FAS, and then I've analyzed the cells. What I'm doing here is not just analyzing one parameter and another parameter, I'm analyzing both of them at the same time. So I can look at the FAS expression inside the CD8 positives or the CD8 negatives. All cytometric data is interrelational. We can look at it relative to itself and to all the other parameters. And this is really, in my mind, the most powerful thing about flow cytometry. We're not just looking at individual characteristics, we're looking at how they interact with one another and relate to one another. Now, this is only a 2-color experiment. People routinely do 12, 15, 16 color. The record is 27 or 28. Mario Roederer at NIH has an instrument that can go that high. And there are technologies for going even higher than that. And we do now have sufficient numbers of known receptors on the surface of T cells, for example, to justify looking at this many parameters interrelationally, where you want to look at them relative to one another. Now, what can we target? I'm sure most of you, if not all of you, are familiar with looking at CD4 on T cells, CD3, cell surface markers, but we're not limited to that, certainly. We can permeabilize cells and label intracellular proteins. We can label things like chaperonins. We can label the intracellular domains of membrane proteins. We can permeabilize the nuclear wall and look at intranuclear proteins, transcription factors. We can label cytokines, chemokines, hundreds, if not thousands, of different targets. And remember, we can do it multi-parametrically. We can look at these things simultaneously. So this is what some of the data looks like. Again, a very simple two-color analysis where we're looking at CD3 versus CD45. As I told you before, we're using light scattering as our initial identification of lymphocytes versus monocytes and macrophages. And by drawing regions we call gates around these, we can then look at the phenotype of these things based on our fluorescent markers. So doing a very simple two-color labeling, we can identify the B cells, the T cells, which are positive for CD3 and CD45. The B cells are negative for CD3 and 45. The basophils, which have low CD3 positivity but are 43 negative. And we can also look at the monocytes and macrophages as well. So this illustrates the multi-parametric nature of flow cytometry. We can go through and look selectively at populations and markers relative to one another. And this can get quite complicated. This is a 9-color T-cell panel here. We've used 9 different fluorescent probes and 9 different cell surface markers. So we have roughly 25 or 30 different 2-parameter plots here. The data can get very complicated. And in fact, now that we're starting to do 12, 15, 18 color, we're starting to rely on a lot of the software and statistical techniques that our colleagues in the proteomics and genomics world are already using to deal with this very high-dimensional, very dense, information-rich data that we simply can't wrap our brains around ourselves. So this is a 9-color experiment, by no means unusual anymore. Fluorescent proteins are a very common target in flow cytometry now. Green fluorescent protein is the one that's most common. Remember, these are expressible markers. We don't tack them on to the surface of the cell. We put the gene into the cell. The gene expresses the fluorescent protein, and that protein can target to the cytoplasm or the plasma membrane or a variety of other areas as well. And GFP, the mutant form of GFP, excites very nicely at 488 nanometer. So it's a great tool for us to use in flow cytometry. And you can analyze it on a flow cytometer and readily identify the GFP positive cells over here. And just like we can do multi-parametric flow using cell surface markers or chemokines or cytokines, we can combine fluorescent protein expression with cell surface marker expression. We can mix these things together and create very powerful tools for analyzing our cells by flow cytometry. There are now a wide variety of fluorescent proteins available in all colors, exciting from the ultraviolet up to the infrared. They are used for imaging, they're used for many other applications, but now that we have multi-laser flow cytometers, we can use these things for flow cytometry as well. And they have many different colors. You can look at multiple expression events simultaneously. This is a 5-color labeling using 5 different fluorescent proteins, GFP, YFP, DS-red, a protein called MCATE, and a protein called cyan fluorescent protein. We needed 3 lasers to excite these, but we can look at 5 different gene expression events simultaneously using our cytometers. And these are commercially available instruments. These are not special custom machines by any means. They are out there. There are also a lot of fluorescent probes available for cell function, hundreds, maybe thousands of them, that target all areas of cell physiology and cell structure. There are fluorescent probes that tag the plasma membrane and can measure plasma membrane transmembrane potential. We can look at ion flux in cells. We can label the mitochondria. We can label the DNA and measure the amount of DNA inside a cell. A lot of tools out there that we can use for flow. Many of you, I'm sure, are familiar with cell cycle analysis. In this figure here, we've taken a DNA dye called propydium iodide, fixed and permeabilized some cells, and labeled those cells with that dye. Using this dye, we can identify the cells that are in the G0, G1 non-cycling phase, the S replication phase, and the G2M phase over here, based on the amount of DNA inside the cell as it goes from 2N to 4N. It gives this nice distribution that allows us to calculate how rapidly the cell is cycling. It's a very easy technique. It's been around for a long time. Still a very powerful technique for measuring cell physiology. And like the GFP and cell surface markers, you can combine this with immunophenotyping. In this case, I've labeled my cells with propydium iodide, but I've also labeled them with an antibody against cyclin B1, which is expressed in the M phase of the cell cycle. It's a cell cycle dependent molecule that excites in the M phase and then is degraded in G1. So the G1 cells are here, the S phase cells are here, the G2S are here, and you can see where the M phase cells are. They have a lot of cyclin B1 expression. So again, we can take advantage of the multi-parametric nature of flow cytometry and combine assays together to get more analytical power. Cell death, apoptosis, is a wonderful thing to analyze by flow cytometry. There are many assays out there for it, many reagents. Here we're using a reagent I'm sure many of you are familiar with, Annexin 5. It labels cells that are undergoing cell death. Their phosphatidylserine residues flip to the surface when the cytoskeleton breaks down. We're able to clearly delineate the viable cells, the early apoptotics down here, and then we've also thrown in a DNA dye that will label the cells where the membrane has started to perforate and the dye can get inside. So we can see whether our cells are dying or not and at what stage of death they are. We can also use forward and side scatter to identify viable and apoptotic cells. Viable cells have a stronger forward scatter signal but a weaker side scatter. When the cells die, they undergo a shift. So this rather simple two-color assay is quite powerful. We're looking at 4 different characteristics of cell death simultaneously using very inexpensive reagents and a very simple flow cytometer. And there are a lot of really kind of cool things we can analyze by flow too. Mammalian cells, human cells are only one thing that we can run through an instrument. As long as we can get something, a microscopic object, into a suspension that will fit through that flow cell, you can run it on a flow cytometer. People have run bacteria, protozoans, exosomes, microparticles, diatoms, plankton, all sorts of interesting things through instruments. Really anything from about 20 micron down to the size of a large virus have been analyzed by flow cytometry. This is a piece of data that came out of Woods Hole Institute. What they are doing is using flow cytometry to look at extremely small marine bacteria in the 100 to 200 nanometer range. Synechococcus, Prochlorococcus down here, which is an algae. It makes up 10% of the carbon sink biomass on Earth. There are huge blooms of it sitting in the ocean. No one knew it existed until 20 years ago. Very, very tiny, less than 200 nanometer in diameter, and yet you can see it on a flow cytometer. The pico-eukaryotes, this is an interesting organism. It's actually a eukaryotic organism. It is the smallest eukaryotic organism, less than 200 nanometer in diameter, was not known about at all until it was seen on a flow cytometer, sorted, and then sequenced. It was something we didn't know about at all. And these are actually some beads on our own cytometer back in my lab that are 200 nanometer in diameter. That's almost the size of a large protein. We can go down pretty small by flow cytometry. People are using flow to look at apoptotic bodies, to look at microparticles, to look at exosomes. There are all sorts of things we can do with this instrument. So I will finish just telling you about some new technology, a trend in flow cytometry that I think is very exciting. Most flow cytometers are pretty big, pretty expensive. They're built with very high quality components. They cost hundreds of thousands of dollars. What we're starting to see now are very small, inexpensive machines. And while many of these machines are simple, 1-2 color analyzers for doing point of care, doing CD4 counts in Africa, things like that, we're starting to see small machines that have the capability of the larger ones, that can do 8, 10, 12, 14 colors simultaneously. And this is one such machine. This is the Beckman Coulter Cytoflex. It was originally developed by Yong Chen. He's a former BD employee who went back to China, started his own company called Cytogen, built this little cytometer, and then Beckman Coulter picked it up. I could hold it in my hands very easily. It's a very lightweight machine, yet it can analyze up to 14 fluorescent parameters simultaneously. It has up to 3 or 4 lasers. It has some very unique technology in it. It uses photodiodes rather than PMTs for fluorescence detection. It uses peristaltic pumps instead of air pressure. It's been built to be small and inexpensive, and it's a very, very nice little machine. This is what it looks like on the inside. It's got tiny little lasers. There's our red, our 488, our violet, our near UV over here. The detectors and filters are located over here, and you can see they're very, very small. He's really compacted the size of this machine down to where it can be easily transported, easily set up in almost any laboratory environment, and it can run up to 14 colors simultaneously, which is something that, until a year ago you had to buy a big BD Fortessa to buy. So I think this is a very exciting trend. This is not the only instrument like this. There are a number of other manufacturers who are doing similar things. I think the machines are getting less expensive, but without losing their capabilities. And it's going to make flow accessible to a much wider audience than it has before in the past. So I just want to end with these comments. Just remember that although flow is mostly applied to immune cells and tumor cells, it doesn't have to be. You can run all sorts of things through a flow cytometer. Any microscopic object is fair game for flow cytometric analysis. And I think it's great that the cost is coming down, finally. It's taken a long time, but we're starting to see inexpensive platforms that don't sacrifice the capabilities that the larger platforms have. So I will stop there, and I'll be happy to take any questions if anyone has any. Sure. Is there a... yeah. Sure, absolutely.
With respect to flow sorting the cell,
Right.
how many fluorochrome we can use without affecting the cells, starters?
Yeah, that's a good point, because cell sorting, when the cell is sorted, it's pressurized. It's pressurized and then depressurized as it comes out of the nozzle. So does this have a negative effect on the cells? For most cells, it probably does not. We found that lymphocytes, most cell lines are pretty rugged, that they can survive this treatment. Some cell types, like stem cells, dendritic cells, however, can be very sensitive. And it is something that needs to be assessed if you're doing a new cell type. Even the ultraviolet lasers on some cytometers can actually cause a little bit of DNA damage as the cells pass through them. So it's like any other technique that you apply to cells. You do have to be cognizant of the fact that it might have some effect. But the effects are pretty well understood. Cell sorting was developed really around the same time that flow cytometry was developed. The early immunologists who were building the first 1 and 2 color flow cytometers, they didn't just want to analyze cells, they wanted to separate them. So the separation technology was developed back in the 1970s, back when the first analyzers were also developed. So it's a technique that's been around with us for quite a long time, and we have a pretty good understanding of it. Yes?
Can we differentiate apoptotic versus necrotic cells by flow?
Necrotic?
Apoptotic versus necrotic cells.
Yeah, that's always tricky because when you induce apoptosis in vitro, the cells go through the entire apoptotic process, and then they just sit there perforated and dead, but they're still intact. Did those cells die apoptotically or necrotically? And it can be difficult to tell that simply by looking at the fluorescence indicators. What I always recommend what people do when they're looking at apoptosis is do time points, because necrosis will usually be pretty instantaneous. If the cells are shocked, physically shocked, and are traumatized and become damaged, that will be a very rapid process. Apoptosis usually occurs over a period of hours. And what I would recommend anyone looking at cell death in a system that's new is to look over time points. And if you're using a drug, do a drug dose curve as well. Physicians often tend to take their knowledge of human dosing and try to apply it to cells, and often wind up giving cells huge quantities of drug that are not necessarily relevant physiologically. So do dose curves and do time curves, and that's really the best way you can determine whether you're seeing apoptosis or necrosis.
I have 2 points. One is the analysis part of it, about compensation issue when you have a more number of fluorescent coming into picture. Right. And second point is cost of machine is going down, but what about the cost of antibodies that are going into it?
Yeah, that's a good point. I didn't talk about fluorescence compensation. I dealt more with the technical issues today. I'm sure many of you who do flow are very familiar with compensation. It's our biggest headache as a rule, because we do have to control for the overlap of fluorescent probes into one another. And that can be done electronically or in the software. It becomes complicated, though, as we start to move into 10, 12, 14 color, because often we have fluorescent probes that are quite close to one another spectrally and require a lot of spillover compensation to subtract them from each other. So it's something that you really need to watch. And if you have a more sophisticated machine and you're doing 6, 7, 8 color, a more intermediate number of parameters, choose fluorescent probes that don't overlap into each other very much. BD has some very nice grids that show the relative overlap, and you can make some intelligent decisions about what fluorescent probes to choose when you're analyzing. And I'm sorry, your second question was? Yeah, I mean I think we're starting to see that there are companies now that are starting to offer the sort of freely available clones at much lower cost than they were before, but the cost of antibodies is still a tremendous problem, and it is a bigger problem the more parameters you add to your analysis, because you're now needing 10, 12, 14 colors to do the type of analysis that you want. I think you're starting to see discount companies, though, that are taking the sort of public domain clones and offering those reagents at a far lower cost than they used to, simply because they can. They can do it in quantity and still make a profit. So I'm hoping the newer antibodies aren't going to get cheaper, but the older antibodies will, I think. Yes? The first question actually involves user-friendliness of both the instrumentation and the software, and the second question involves challenges of live cell sorting. I think one thing that I've been pretty impressed with some of the newer, smaller machines is that the developers, rather than try to reinvent the wheel, as we say, they've gone to the packages that we're already familiar with DIVA, with CellQuest, and borrowed heavily from them, which is all to the good. because it makes these instruments much easier to run. There is always a learning curve, but I'm actually pleased with the direction that software is taking in the smaller instruments, and I think we're very fortunate in flow cytometry to have very good third-party analysis packages like FlowJo, FCS Express from De Novo Software, Verity Software House that writes ModFit, and WinList. These are all well-written packages written by people who know flow cytometry very, very well, and they compete intensely with each other. So when a new machine comes out, they immediately race over to that company and say, give us some data from your instrument so we can make sure our package can analyze it. So I think that's something we're very fortunate about in the flow world, that we have pretty good third-party packages. There are also, there's always been a small amount of free software out there. The flowing software package out of the University of Turku in Finland. What are some of the others? There's a couple others as well. WinMDI is older. WinMDI won't run on Windows 7. It's a 16-bit. But there are a couple of other free. There's always been a community of software developers out there that have provided simple free software as well, and that can be very helpful as well. In terms of sorting live cells, I run a sorting facility. Sorting is 90% of what we do, and it's like anything else in biology. It's 90% sample and 10% instrument. You know, you want to provide cells that are healthy. You want them to be in good media. You want to sort them into the media that they're comfortable with. You want to make sure that the PBS that you're using as sheath buffer is HEPS buffered so that the pH remains constant. You want to process the cells promptly after sorting. Most of the problems we've seen with viability and sorting are not related to the sorting itself, but just because the cells are unhappy at some point in the process before or after sorting. So really pay attention to these things. We have people who sort 100 million cells and they don't do anything with the cells until the end of the sort 8 hours later. The cells have been sitting for 6 hours at room temperature in PBS. So no, they're not going to be, they're going to be dead. And it isn't because the sorter made a mistake, it was because the cells weren't handled properly. So really pay attention to these details. Yep.
Thank you. Thanks a lot.
We'll
have enough time.
Yeah, we'll all be around afterwards, so please come up and talk to us.
We want to have enough time for speakers. Yeah, at the end of the talk, all the three talks, then we'll have enough time to have one-on-one with them.
So, thank you very much, Dr. Will. I feel uncomfortable in breaking the interesting discussion, but we have to move on. Next, I call upon Dr. Vivek Tanavde, who is Principal Investigator at the Institute of Medical Biology at A-STAR in Singapore. He is the sequencing man, and his topic today is the sequence century, how sequencing is transforming healthcare. And Dr. Vivek Tanavde, please.
Okay. First, I'm going to introduce some of the current sequencing technologies. I'm not going to talk so much about what's coming in the future because that's still some time away. And then I will talk about the second part will deal with some specific examples that we've done in my lab. And these are also structured into 3 parts. Okay? So the first part will be something basic that has potential in healthcare. We don't know that yet. The second is something more applied. And the third is a disease, because Dr. Chirag Desai is here. And we have to talk about cancer, so I'm going to talk a little bit about glioblastoma. Okay, so the whole idea of the current what we call as next generation sequencing, and there's a debate now whether we should even call it next generation because it's already current, so we should call it current generation sequencing, not necessarily next generation sequencing. But this is built around the idea of massively parallel. So this is built around the idea of massively parallel signature sequencing. So essentially what this technology does is that it can count individual RNA molecules. Okay? And that's really the simple part There's nothing else more to it. Now it can do this. It can count molecules that are in solution. It can do this why massively parallel, because it can do this millions of molecules at a time. Okay. Now, one big difference, some of you are familiar with microarrays, which are probe-based or any other probe-based technology, is that in probe-based technologies, you only see what you're looking for. So if you're looking for a particular gene or a particular probe, you see that. You have a probe for that, you see that. In this kind of technology, you see everything. Okay, what you're looking for as well as what you're not looking for, because it doesn't care. It doesn't really take a probe there. Okay. Now, as I said, a library can have millions of individual tags. There are two major challenges in this field when you count this millions of tags. One is linearity. So, if you counted 10,000 tags versus 100 million tags, would you get the same kind of results? Okay, and the answer is no, but there are ways to deal around this. I won't go into much detail about that. But you can still scale this up to a large extent. The other is sensitivity. Okay, now the challenge of sensitivity here is not that this method is less sensitive. The challenge is that the method is extremely sensitive. Okay, so in theory, you can actually count even a single molecule of RNA or DNA that is there. And the challenge then becomes, how do you assign meaning to this? Okay. Now you're in a position where you can really fish out that one DNA or RNA molecule, but then how do you interpret this? What do you make out of it? So the linearity challenge is solved by amplification, and from a single molecule or a single bead, I'll cover about how exactly this is on the Illumina system, and different chemistries do this slightly differently. But basically, what you do is, no matter what kind of technology you're doing, you have to amplify. Now, the amplification linearity, again, is not necessarily linear. The more you amplify, the higher is the error. And we were discussing this yesterday. So in theory now, you can do this from a single cell. Okay? You can do sequencing from a single cell. Of course, when you do that, you need much more amplification than when you had 1 million cells. Okay, so if you have enough from 1 million cells, but when you prepare libraries, you're always amplifying. Now, the advantage here of amplification, you have to remember, is that this is not like the PCR amplification. Okay, this is just doubling in every cycle. This is not a geometric progression. So even though we say amplification, this is actually, the error here is lesser because the amplification is lesser with the same cycles compared to your traditional PCR that you do. Okay, so essentially, now what we're doing is, again, the difference from an array is in a microarray or any kind of probe-based technologies, we were looking at a signal, and you're saying that the signal is directly proportional to the amount of DNA or RNA that is in your sample. Okay, whereas here we are directly counting molecules. So in a way, this is a digital technology rather than an analog technology. Okay? Because we are directly counting the number of tags, the number of molecules, and that's why we can get pretty accurate results. Okay? So the current Illumina technology, now I have no affiliation with Illumina, but of the technologies that were introduced, this is one of the only ones that has really survived and continues to progress in time, because initially there were 3 major technologies: Illumina, Solid from ABI, and 454 Roche. Solid is discontinued today. The platform doesn't exist. The machines do, and they're supporting it, but they're not making any more new machines on the Solid platform. ABI was taken up, merged with Life Technologies, which then was bought over by Thermo, and so that platform doesn't exist. 454, the big attraction was that you could get long reads. And so the throughput was not very high, but you could get longer pieces of DNA or RNA, and so that's why that was the attraction. But today this is no longer the case because the Illumina machines have caught up, and there are other technologies which I'll talk about which are upcoming, which can give even longer reads. So there's really no advantage to using 454 today. And that's why you don't see a lot of those machines being sold. And so this is really the dominant technology. And essentially, what it does, it says this is sequencing by synthesis. So what happens here is, this is true of the chemistry is different, but the overall procedure remains the same. You take your DNA or RNA here, and you fragment it into small fragments. Now, there are a number of ways in which you could fragment this. You could use a shearing machine, like you could use a mechanical shearer. Okay, so basically, it's just chopping this into small pieces mechanically, like a blender. Okay, or you could use ultrasound to chop this, or you could do this enzymatically. You can use a restriction enzyme that will cut this piece of DNA into small DNA pieces. And which method you choose really depends on your cell type, because the DNA and RNA composition is different in different cell types or even different organisms. And so what works best for one may not necessarily work very well for the other. Yeah? And so you would have to do some trial and error and pick the shearing that works best. Now, okay. So you have these fragments which are essentially a couple of hundred base pairs, and then you add adapters, okay, and then you apply them to a flow cell that looks like this, and this is called as the procedure here is called as cluster generation. So essentially, you have a PCR, but as I said, this is a PCR where the amplification is not geometric, and then you amplify this using a bridge. So why do you need adapters? Because on both ends now you can have sticky ends. And so you can put stuff there that it will stick to. This is solid phase sequencing. This whole thing happens in the Illumina system on a bead. And you have a large surface area to a very small volume. So you can literally do a lot of such molecules in parallel. Okay? And then you put this into a machine, and you essentially get the final output here is an image like this, okay, which is read automatically by a computer in real time. And each nucleotide has a different color. And so you essentially, Bill talked about 16 colors. Here we need only 4 colors, okay, because we have only 4 nucleotides. And so you essentially have a 4-color experiment where each dot is read by the computer, and that's how you get your sequence. Okay, so that's how the Illumina system works currently. And when this, the system itself, like flow cytometry, is also evolving over time. Okay, so what we had in the beginning was a machine which would basically do 200 megabases of data. The first Solexa that was developed by a company called Solexa, and Illumina bought over the company. And then came a machine, what they call as the high-seq, because it was highly efficient in sequencing, and then they call it the high-seq 1000 because it could do 1000 base pairs. And then came the 2000, then they came 2500, and today there's a number of other, they call it next-seq. Okay? And essentially what has happened is that the chemistry hasn't changed all that much fundamentally, but the capacity has got more. And so where you could get data, where you could sequence maybe 200 megabases, now today we can do, 5 or 6 billion bases, 5 gigabases today, or 6 gigabases today. And so what that led to was a massive reduction in the cost of sequencing. And here what you see here, this is a slide I borrowed from NHGRI. You see around 2007, okay, was the inflection point. So until this, and this is when the first Illumina machines really were sold in bulk. Okay, 2006 was when they launched, I think, the high-seq machines, and then the scale for the high-seq. In fact, when they launched the high-seq 2000, they said we're launching this machine, but if you want to buy one today, there's a 6-month waiting period. There's a wait list. Okay, and the reason for that was the day they launched this, or before they launched it, there was a company in China called as BGI, Beijing Genomics Institute, that bought 126 machines, okay, before anybody else, just because of the capacity of sequencing. 100 of these machines were to be stationed in Hong Kong, and 25 were going to be in Shenzhen, in China. Okay? And so when these machines started coming online, the cost dropped dramatically. And as you can see, this is under 2014. It's starting to plateau off now, but this is actually dropping faster than Moore's law. And what is Moore's law? Moore's law is a law that predicted what would be the cost reduction in computer chips. So we know that my phone today has more computing capability than a desktop that I wrote my thesis on. Yeah, and it doesn't cost as much. right? So this is what Moore's law predicted, and this is the slope of Moore's law, and you see the cost per megabase for sequencing has dropped exponentially. And so what does this mean? What this means is that this is becoming cheaper and cheaper, and more people are going to use it. And today, the limitation is not really the hardware or the cost of sequencing. It is the data that's coming out of it, and how do you manage and analyze this data. So what are the barriers? So why isn't everybody in this doing sequencing today? Right? If this is getting so cheap, if this is so applicable, why isn't everybody doing this? So the first barrier is still availability of NGS platforms. And again, here the model is changing. Okay? So initially, it was very few places had it. And so this is like a Mercedes or a BMW. Okay? People will put this on their website saying, we have an Illumina, iSeq, whatever. Okay? And because very few places had it, they controlled this, and very few places can use it. However, today, if you see, there are companies, and there are companies in India as well, there's a bunch of them in Bangalore that I'm familiar with, which will basically do this for you. So we're going to a model, it is very similar in evolution to, if you think of the computing industry, in terms of what PCs did to mainframes. Okay? In the past, we had big mainframe computers that did all the work. And you had clients that you just had to log into. And what really changed is the development of the PC and when Windows came online. And suddenly everybody had a computer in their home, and you could do things without logging into the mainframe, because to access the mainframe, you actually had to go to the office. Right? So this is something which is similar that's happening in NGS. Second is cost. It's still expensive. It's not cheap, but as we saw, the cost is coming down, and so at some point it's going to get actually cheaper than microarray. Okay? In fact, my lab works a lot with microRNAs, and quite a few years ago, about 5 years ago, Illumina stopped selling microRNA microarrays. And when I asked them why, because I really liked that platform, they said because everybody is going to do sequencing. And the cost for sequencing is going to be the same or cheaper than that for microarray. So why would you need microarray for microRNAs? And this was especially true for microRNAs because these are small. Right? So your cost of sequencing is not very high, because cost per megabase, but you only need to sequence less than 100, 35, 50 megabase base pairs. Okay? Then the infrastructure for library preparation. And again, this used to be, in the beginning, something very, very expensive and very specialized. So I told you, now imagine you have to try out 3 different sharing methods. Right? But over time, the cost of these equipment has also dropped down. And so you can now have 2 different equipment for sharing in your lab without spending a lot of money. Okay? And of course, the last point still is valid to a large extent, and that is how do we deal with the data. Right? So today, this is a real bottleneck, because I can send my samples to Bangalore, okay, and they will sequence it, and I can write a grant and get money. And it's not even now. I think the cost is actually probably under $1 lakh per sample, if it's depending on what your sample is. But so I can spend a few lakhs and get this data done, right, from a grant. But then what do I do with the data? And we're trying to address that by having courses which actually teach you data analysis. Some of you have participated in these courses, right? So that's kind of where we are today commonly. Okay? But this is also changing, and that's another challenge, is that the technology is changing very, very fast. Okay? And where are we going? We're going towards, so one limitation of the massively parallel sequencing that I spoke about is that you need to shear your DNA into small bits and fragments. Then you sequence these fragments and you assemble them back with the help of a computer. But this also introduces errors, and so that's something we wouldn't want to do. And where the field is moving is towards single molecule sequencing. And these are the 3 technologies. And as you can see, one is already bankrupt, okay, in this field. So the first is Pacific Biosciences, which is probably the most mature technology. They call it single molecule real-time SMRT technology. The second is Oxford Nanopore, where it's becoming very, very exciting, and I'll talk about this in the next couple of slides. And third was Helicos Biosciences, which was a company that came out of Stanford. Stephen Quake but they're already bankrupt. But essentially what they did was they've used this technology, not in the commercial setting. The company went bankrupt, but they've used this technology to in the clinical setting to a large extent, and probably this will get morphed into the technology will be bought over by some other company who would still use this technology. So, as I said, PacBio is the most mature technology. Now, this is the cartoon on the right. So, essentially, what you see here is that this is still fluorescence based, and this is one of the limitations of many technologies. The moment you have fluorescence, you need lasers, as we saw in the previous talk, and you need detectors and PMTs or CCDs, or these can be expensive and they make the machines bigger. Okay, but PacBio is still fluorescence based, and the error rate when they began was very, very high. In fact, the company would not sell you the machine. Okay, because you then would claim that if I buy something, I wanted to perform as advertised. They would say that you are a collaborator and you pay us the money for the machine. So you're buying the machine, but technically they're not selling it to you. They are collaborating with you on this project. Okay, but they've made tremendous improvement in this, and today's error rate is less than 0.01%, which sounds very good. But remember, if we're talking of billions of base pairs of a human genome, in absolute terms, that error is still relatively high. But we're getting to a point where PacBio is now becoming very comparable to the Illumina system, which is really the standard that people use. And the biggest advantage is long read length, as I said. So essentially you get something like the median length, as you can see in the graph on the right, is around 14 kb. So we're going from a few hundred base pairs on the Illumina system to 14,000 base pairs. And the longest ones are over 40 kb. And so this is something which is already in the market today and pretty mature. But what I'm really excited about is this technology from a company called as Oxford Nanopore, which is called as nanopore-based sequencing. And this is again a platform that is really evolving tremendously. Okay, there's a lot of work that's been going on in this area, and Oxford Nanopore was the first company, but by no means they will be the only company doing this. And essentially what this does is that the idea here is very simple. You have a pore, a nanopore, so it's nano-sized, and it's so small that at one time only one nucleotide can go through this. Okay, much like a sorter that Bill showed. So you have like a flow cell. So you see the pore here, and this nanopore can be a protein, it can be a salt compound, or it could be actually even a nano compound or material. So that's one area where there's a lot of innovation that's going about, because the advantage of protein is that proteins are very reproducible in forming nanopores. This is their natural structure. Okay? The disadvantage is that proteins are unstable, and so you need more stability. But they're cheap to manufacture. You can make tons of protein, and we know how to purify them, we know how to crystallize them, we know how to put them on any kind of surface. Whereas other materials are more stable, but they're more challenging in terms of applying this. Yeah? But essentially what you do is you have a nanopore here, you allow your analyte to pass through, and then what you do is you measure the current. Okay? So the big difference, or the big innovation here, is that we are getting rid of fluorescence. So because there are no fluorescence detectors, the machines can be very compact. And you don't need detectors, the cost comes down tremendously. In the Oxford nanopore system here, and a lot of other nanopores, you detect current, okay, or changes in current. So what you see here on the left is that if there is no analyte passing through, you get basically this stable current flow. Okay? And now if you have different size analytes or different chemical compounds of analytes, then you get different patterns of disruption in this current. And this pattern can be specific for your A, G, T, or C base, and that's how you detect this. Okay? Now, the other exciting thing about the nanopore is that nanopore doesn't really care whether it's a nucleic acid or anything else, a metabolite. Okay? And so you can basically take this kind of similar technology and apply it to other things. It doesn't have to be restricted to only sequencing or nucleic acids. The other field where a lot of development is happening is in the area of detection. So I told you that there are disruption in current, but there are multiple ways to really detect these disruptions. And today the sensitivity is so great that you can measure basically a single hydrogen atom change can be measured in this. And so you can actually measure compounds that are having differences in structure using the nanopore technology. There's a lot of work that's going on in this area right now. And this is the evolution, right? So the Illumina machines are big. They occupy about half this space. And this was a company called Minion, and they were bought over. They're bought over by Oxford Nanopore because this is also built around nanopore. And essentially what this is that this is a USB size sequence. It's a thick USB thumb drive. Okay, it's about twice the thickness of a regular thumb drive. But basically, the whole advantage of this is that you can plug it into the USB port of your computer, as it's shown here, relative to a computer, a laptop, right? The whole chemistry for extraction is on the chip. So you can directly add your blood, plasma, whatever is the fluid, and it gets powered. It has small micromotors inside that power this. The extraction of DNA or RNA is done on the chip. It goes into the microcapillaries where this is run, and basically the data is output directly to your computer. Okay, and the software to analyze the data is also on this same chip. In fact, just about a week ago, they came up with, there was a consortium. So there's an early access program. Now, this thing costs, I think, about $1,000 US. That's it. Okay? Some of our phones cost about that much. Right? And they have this early access program, so you can apply for this access and get this. And there's a user group or consortium, and they released the first results of a large-scale application of this kind of technology. And the error rates are still not as good as Illumina. They're relatively high, but still we're getting there. They already have the Mark 1 version of this. This picture is the original one. Now they have a Mark 1 version. I'm sure they're working on a Mark 2 version. And as this technology develops, it's going to get cheaper and better. Right? Now the limitation as of now of this is that it's still limited to about 14, 15, 20 kilobases. So it's not single molecule, but it can't really handle very, very long. But again, this is an area which is undergoing improvement, and so the newer versions are going to be able. The goal is to have a complete molecule sequenced without any fragmentation. Now, you might ask, why is this necessary? Okay, or what are the real advantages of, or why are we going towards single molecule? So there are certain applications where single molecule really gives you an advantage. Now, whenever you think about any technology, it's always a mature technology that works really well, the efficiencies have been worked out very well, and that is cheap. Okay? Versus something new that can do something better, but doesn't work as well all the time. And this is always a conundrum. So the Illumina technology is very mature, works extremely well, guaranteed to work, and the cost per megabase is relatively cheap, or the ion torrent or any of the other technologies. Whereas something like this is not that mature, and so the error rates are high. It's per MB, the cost is more. But when you think of applications like splicing, okay, novel splice junctions, it's very difficult to detect this by parallel sequencing because you have two pieces of DNA, okay, or you have two pieces of RNA, and these are joined. Okay. Now, what if while you're shearing, this got sheared at exactly the same point? Now, unless you know that this was the splice junction, you are not going to be able to identify this reliably. But in a single molecule sequencing, you don't have this problem. Okay. And so there are areas like detecting splice junction where this kind of technology gives you a real advantage, and that's probably where it's going to be used first. Okay. Okay, so that was the first part of my talk, and this kind of summarizes the slide where we say that today everybody is doing GWAS studies, genome-wide association studies using NGS. Okay, people are doing exome sequencing, which is cheaper because you don't need to sequence everything, but on a large sample size. And there are already the 1,000 genome projects, right? That take, in fact, there's a 1,000 genome project for every disease now in the US. So they've identified some 17 or 18 diseases and 1,000 patients defined, and all this data is going to be publicly available. And so anyone who wants can look at this data. Okay, we can do this. For example, we had in India a project on tuberculosis, which is open source drug discovery, right? It could be open source genome analysis. Once the data, and today almost every journal wants you to deposit your data in a publicly available domain when you publish your paper, right? So there's a lot of data that you can access publicly and play around with. If we understand the complexity of gene regulation better, then we can have new therapeutic targets. Epigenetic is a good example of this. In cancer today, there's a trend of HDAC inhibitors with classical drugs give you much better disease management. And that's because we understand that the regulation and the targets work differently, and you can have drugs that target both these. Okay? And if we have more precise measurement of all these analytes, it then gives us a better way to model, because one of the limitations of modeling, and I work at the Bioinformatics Institute, and I have an affiliation with the Institute for Medical Biology where we use some of these things, is that our measurements are not very precise. And if your measurements are not precise, your model is never going to be very good. And that's why we don't really have theoretical biology, unlike theoretical physics. Okay? Because in theoretical physics, we can make pretty precise measurement in physics of, if there's an electron that's flapping, you can measure this pretty precisely. And you can then develop a quantitative model very precisely. In biology, we're not there yet. But if we're able to more precisely measure, then we'll be able to more precisely look at this and develop this. Okay, so now I'm going to tell you 3 stories in the next couple of slides. And these are stories that my lab has been involved in. The first one talks about this fish, okay? And this is a prelude to the cockroach. And you'll know what a cockroach is when Rams talks, okay? So we work with this model called a zebrafish, and it's a very good model, and it's a model for disease, and that's why I put it here, because increasingly there are a lot of diseases from even breast cancer, okay, which is modeled in zebrafish. You can develop breast cancer in zebrafish. The same genes can be expressed, you can see similar phenotypes. So it's a small fish, you can culture these fishes, you can grow them in large numbers, you can do all sorts of experiments with it. The genetics has been worked out very well, so it's a good model system that people are using for disease. The collaborator I'm working with us on this, Mathavan at the medical school, which is next door to us, is now working with the Shankar Netra lab in Chennai. And so they are looking at using zebrafish as a model for eye disorders, okay? Because zebrafish also has an eye, and we also have an eye, and so they're looking at eye disorders with Shankar Netra. So it's a very interesting model. And what was really surprising was that the microRNA profiles of zebrafish wasn't very complete. So when we started this work, there were about roughly 600 to 700 known microRNAs. And there have been 2 builds since then, and this has increased to about 1,500 in the latest version, right? But this is still a small number compared to what we see with humans. So essentially, the project was very simple. This was more technology but with the aim of understanding the biology. Okay? And we're not understood the biology at all. The first paper was just the technology part. So what we've done is we've sequenced. So we took different tissues of zebrafish, sequenced them so these are all millions of reads. Okay, we use the Illumina HiSeq platform, and the idea was to compare all this to embryo. And why embryo? Because my collaborator was interested in asking the question, what happens during development? So now you have an adult tissue, of brain, testis, heart, etc., and you compare it to an embryo, so you can do 2 things. One is, instead of taking a whole fish, you can actually take a whole fish and grind it up, okay, and take the RNA, and that becomes one control. But that's developmentally mature. But if you take the embryo, then you can also look for genes that are changing as an organ develops. And that was the rationale for taking an embryo. And I won't go into details of the other stuff that we did. But essentially what we did was we were able to identify almost more than 1,000. So at that time, remember, 600 were known. But we got about 1,500 novel microRNAs. These are novel predicted microRNAs in zebrafish. Okay? And so we're almost doubling the size of the zebrafish mirror. Now this has just been accepted last week in BMC genomics, and so we've deposited data with EBI. There's a database for microRNAs called Mirbase, and once the paper is out on the website, then this gets officially incorporated into Mirbase. Yeah? Now, what you see here is that, so using a technology like this, we can identify novel microRNAs. These microRNAs could be important in tissue-specific. Now, these are all tissue-specific microRNAs. Okay, so they could be important because they are expressed only in that tissue, in regulation of tissue function. And if you're understanding disease and trying to use zebrafish as a disease model, then knowing this becomes important. The other thing is that now we are in a position to link some of these to the humans and ask the question, what are the human homologs? Okay, so what I would like to really point out here is that if you look at the distribution of microRNAs, novel microRNAs, you see most of these are in the 1 to 100 range. There is 1 to 100 transcripts or below. Okay? And that's probably, there are very few in, this is L5 is 10,000 to 100,000 transcripts. There are very few there. Most of them are below 100 transcripts. And the reason why they were not discovered so far is because they're expressed in such low concentration. Okay? They're present in such low numbers. But doesn't mean that they are any less important, because even if you think of genes, like transcription factors like P53 or MYC are not expressed in very high amounts. But a slight change is enough to basically change a cell from a fibroblast to a stem cell. Right? So the reason why these were not discovered was because they're expressed so rare. But the platform like this can actually pick these up. And we can now, as I said, the sensitivity is even single molecule. So we can start to study these rare microRNAs and ask questions of what genes do they regulate, what functions do they have, how are they themselves regulated. There's a whole lot of questions that we can ask. Okay? So that was one story. The second story is what is my core interest of my lab, and that is understanding mesenchymal stem cells. So that's what my lab works on. And these are stem cells that are found in bone marrow, dental pulp, adipose tissue, all kinds of places, right? Now, the challenge really has been to understand how differentiation works in these cells. And can we get targeted differentiation? Okay. So, yesterday I was talking to BK and he said what he really wants are replaceable body parts. I need to get my spine oiled, okay? Much like a car. And Bill said that the recent flow cytometers are like Volkswagens, and you can change parts or swap parts, right? Now, to get there, first we need to be able to swap cells. Okay, tissues are more complex. To basically take out a liver and put a liver and swap it in, that's more complex. But if I can, first step is I need to look at cells. And the goal of my lab has been to get targeted differentiation of cells. That means, now these are the three endpoints we study, okay? Fat, bone, and cartilage. These cells are capable of doing all these three, getting into all these three different endpoints, but you don't want to have all three endpoints in one place. You want to have only one endpoint. Because, for example, if you want your cells to make fat under the skin, you don't want to have bone there. That's a bad idea. Okay? Or if they're in the cartilage, in the chondrocyte, in the knee joint, again, you don't want to have bone there. So one way of understanding this was to look at expression data. And this was done by microarray at that time, but essentially we could do a similar question. You could ask similar question using sequencing technologies today. And today I would do this using NGS. So what we did was we looked at gene expression data, and we developed methods to correlate this with phenotype, to correlate this with looking at the expression, can I tell something about the cell? Okay? And by using this kind of approaches, we were able to identify three pathways, okay, which are specific for each lineage. And what happens, and that's what I'm showing here, is the first one is this is the top panel is the control, so it's normal differentiation. Okay, and each of these three molecules inhibit specific pathways. Okay, one of them, the tyrosine, is a PDGF receptor inhibitor. The SB431542 is a TGF beta inhibitor, and the SU5402 is an FGF inhibitor. So and the nice thing is that these are all failed drugs. Okay, they were at some point proprietary molecules developed by the drug industry didn't make it because, of course, if you inhibit any of these pathways, it's going to be very toxic. But now you can buy them from Sigma or CalBiochem. And in the concentration that you use, they're very, very specific. Okay? And so what you see here is that if you inhibit, for example, PDGF signaling, I get basically a slight inhibition of fat, I get very little bone, and I still have cartilage. Whereas if I inhibit TGF-beta signaling, I get more fat. Okay? And I get good bone, but no cartilage. And if I inhibit FGF signaling, then I get no bone at all. So if my goal is to inhibit bone formation, all I have to do is switch off FGF. Okay? And I'll get no bone. Unfortunately, the reality is not so easy. And so you can do that, and this is example, but essentially this comes at the expense of growth. Because again, in biology, things are very, very complex and interrelated. So these three pathways which are critical for differentiation are also very critical for growth. Okay? This is in the same cell system. When we started looking, so we started asking the question, how does this happen in vivo? And the answer was that in vivo, microRNAs regulate these processes. Okay? And the answer, it's not an on-off switch, but it's a fine modular, fine tuning. And so we started looking at microRNAs, and this is an example of where we did sequencing, or my collaborator. It's even better when somebody else pays for the sequencing and you analyze the data and publish the paper. Okay? That's better than paying for it yourself. So I had a collaborator who was interested in asking the question, what are microRNAs inside the cells and outside the cells? And how do these change? Okay? So we answered that question for them. But after we did that, we said, we have the data. Why don't we also ask our own questions? And our own question was, are there any networks of microRNAs that regulate gene networks? Okay, that would then regulate a physiological process. And so when we started looking at this data, we generated now, since we had the sequence information, we were actually able to generate networks using two different approaches. Okay? On the left, we utilized the sequence information. I won't go into details of how we did that. But we utilized the sequence information to predict targets. And we knew that we are under-predicting. Okay? We said that's fine. And then we used target scan, which is also a sequence-based prediction algorithm, but it massively over-predicts. You get a lot of false positives. You get only about 20 to 30% of your actual results are real. Okay, 70 to 80% false positive. So it massively over-predicts. And then we combined an under-predicting method with an over-predicting method and said, can we now achieve something which is more important or useful? And when we looked at networks in both these, there was only one network that was common. And this was a network at the node of which was this transcription factor, HNF4 alpha. Now what's very interesting about this transcription factor is that this transcription factor is not under microRNA control at all. Okay? If you look at the sequence, that is what it will tell you. If you look at the literature, that is what it tells you. And yet, when we were looking at levels of these LET7 family of microRNAs and this transcription factor, we found everywhere a very strong inverse correlation. Okay, so in liver, for example, this is very important for liver hepatocyte differentiation. So when we were looking at this in liver, it has tons of HNF4 alpha. If you block HNF4 alpha, you can get no hepatocytes. And the LET7 was very, very low. In MSCs, LET7 was very high, HNF4 alpha was very low. When MSC is differentiated to liver, that's another advantage, that we can take these and make whatever we want. So we take MSCs and we differentiate them into hepatocytes. You can see the LET7 coming down, HNF4 alpha going up. So we see very good correlation. But every paper that you see, there's nothing that says that these are HNF4 is under this control. And what was actually happening was, this was true. It is not a direct regulation. And that's why we wanted to look at networks, because this was under indirect regulation of the HNF4. HNF4 alpha was under indirect regulation of this microRNA family. And so basically this microRNA, or this family of microRNA, controlled all these genes around it, which in turn regulated HNF4 alpha. So the lesson we learned was not only direct interaction, but indirect regulation and interaction is also very important when we look at microRNAs. And here we wouldn't have been able to do this if we did not have the microRNA sequence. Okay, because if we didn't have the sequence, we would never be able to generate the network on the left. The one on the right would be lost amongst 30 other networks that were generated, and we would never have been able to get this relationship or understand this relationship without the sequencing data. Okay? So again, MSCs are clinically important. They are used in a large quantity of trials. My interest is more in orthopedics, in bone and joint reconstruction, and orthopedic disorders like osteoporosis, but they are being used even for a lot of vascular disorders. People are using them in Crohn's disease, in chronic limb ischemia. There's a company in Bangalore, Therapeutics, that's doing a lot of trials in this area. So people are already putting these into different parts, including the heart. There was a lot of studies where people took MSCs and put them in the heart in acute myocardial infarction. And the scary part is that we don't really know how these work. We understand very little of the biology. And we hope that we put them in the right environment, they don't form the wrong cells. But this was also proven to be wrong, because what was found was a report that initially when people put them in the heart, they said they don't become fibroblasts. Okay? They will all only become cardiomyocytes. Then people found out they don't become cardiomyocytes also. And they said, oh, they actually become blood vessels. They stimulate the existing cardiac muscle cells. That's how they act. And then suddenly there was a paper that said, if you do a large enough study, they found that there were 2 patients where these just became fibroblasts and caused fibrosis. Now you don't want to do that in acute MI heart, right? And so we understand very little of how these really differentiate, but by understanding the mechanisms that regulate differentiation into a particular lineage, we will be able to control this better. Okay, and so the last story that I'm going to talk about before I end, I have about 4 minutes, is directly clinical. Okay, so this was in collaboration with a collaborator at the Institute for Medical Biology, Prabha Sampath, and she was looking at glioblastomas and gliomas. And we were looking at translation. What are the RNAs that are differentially translated? And while we were doing this, and we were using a very age-old method for this, which is density gradient centrifugation. Okay, you have a sucrose gradient, you put your RNA on the top, your soup on the top, and you centrifuge it, and you basically, the assumption is that if it's getting translated, you have a lot of ribosomes, it's very heavy, cells dissolve faster. If it's not getting translated, there are no ribosomes, they stay on top. Okay, so there's a newer method called RiboSeq that is available, but this wouldn't give us this kind of data. So new methods are not always more desirable, right? So we were looking at what is translated, and when we were looking at this and looking at transcript abundance, okay, and whether it's transcript abundance in the neuronal stem cells from the patients, okay, this particular microRNA stood out, MIR138, okay. So this is very, very abundant in the glioblastoma stem cells, but this is not very abundant in the normal neuronal stem cells. Okay? And so what they did was so this was our part in identifying this microRNA. We picked it up because it was not getting translated, because the microRNA doesn't get translated. So this method actually can pick up a lot of non-coding RNAs, including microRNAs. Okay? And essentially then when they started looking at this, they found this very interesting, that if you were to knock down this microRNA, it inhibited neurosphere formation. Now, neurosphere assay is an assay that is used for looking at nerve cells. Okay, so what you see here is on day 0, you put these cells, and these are fluorescently labeled so that you can see them easily. You can even sort them using a flow cytometer, and you see that as time progresses. So this is day 1, day 3, and day 9. By day 9, you get these very nice neurospheres, and this is a functional characteristic of neuronal stem cells, that they can form neurospheres. Okay. Because we use CD133 as a marker to isolate these cells. Now, it's controversial, but they can form good neurospheres as a functional assay. Now, if you inhibit this MIR138, then you basically see no neurosphere formation. Okay. However, and this is shown quantitatively in the next graph here. Okay. Now, the interesting thing was that this is specifically only happens in glioblastoma. Normal NSCs are not affected at all. And subsequently, I'm not showing that data here. So the last graph shows you these different lines. And you see the blue is the scrambled. And they continue to grow. Whereas if you inhibit 138 in these, they stop growing. Okay? And then subsequently, they did animal studies and showed that in a rat brain, you induce glioblastoma in a rat model. And you can inject these antigomirs directly into the brain. And these rats survive longer. And then they did imaging and showed that there is no glioblastoma formation here. So it's very specific for glioblastoma. And glioblastoma is a tumor which doesn't have a treatment. I mean, which is not easy to treat. Probably Dr. Desai would know more about this, but as far as I know, there's no effective treatment here. You can only manage the disease to a certain extent, but you don't really do anything to cure it. And here's a microRNA where if you inhibit this microRNA, you basically inhibit growth of the tumor. The tumor just doesn't grow. Okay? And it's not toxic to the normal cells, which is even better. So this is not currently being developed for clinical trial. We're doing a lot of preclinical data with this by my collaborator. And this was the last example where, again, without a sequencing like technology, without the ability to look at these sequences and to measure transcript abundance very, very precisely, we wouldn't have been able to identify this kind of microRNA. And this is now directly translatable into healthcare. Right? So, in summary, this technology, even today, is becoming easier, clinically more relevant. I have not spoken of other things that not my lab, but some of my colleagues do. A big effort is biomarker development. Again, NGS is being applied for biomarker development, especially in the field of cancer, very, very exhaustively. So, this is really going to revolutionize healthcare. In terms of what kind of information we're getting. And once we have sequencers like the Minion, basically you can do it in any path lab, where they'll be able to generate the data. Now the challenge of analyzing the data and interpreting it still remains, and the only solution is that more and more people are trained how to kind of analyze and interpret this data. Okay, so I will stop here, and I think we can take questions after Ram's talk, right?